Welcome everyone on this EEG data acquisition and pre-processing course. So in this introduction, I will set out the scene. Why are we going back to basics and how these affect reproducibility? As many of you know, EEG is almost a century old. From surveying the literature, we can see it was on the rise pretty much linear until the late 70s. And with the advent of cognitive neuroscience, it's been exponentially increasing since. With increase and with this rise, we see new technology emerging, in particular brain-computer interface and wearable devices. And with increased usage come increase in analytical complexity, which hinders reproducibility. Now, with complexity comes interdisciplinarity. Neuroimaging is a prime example of this. So as illustrated here, EEG and MEG research intersects with many fields, such as engineering, physics, mathematics, and of course, neurophysiology and psychology. So in response to this and the need for clear scientific evidence, OHBM has been developed the Committee on Best Practices and Data Analysis for Sharing. Now, there are many useful standards which have been developed. Standards for sharing, of course, with the brain imaging data structure, but also standards for analysis and reporting with the International Federation of Clinical Neurophysiology, and of course the OHBM COBIDAS. Together with these recommendations come other recommendations, which are about using workflows and pipelines. So if we look at what a workflow is a sequence of tasks to process the data. And that would typically be what we write in the method sections. We cannot, however, report every single parameters. And this is why we typically recommend to also share pipelines. So data analysis pipeline is a script to execute a chain of commands, and this allows to avoid human errors and increase reproducibility. So a typical workflow for EEG analysis would start by identifying the bad channels and removing channels with strong artifacts, followed by some filtering, the eliminations of physiological artifacts, and then re-referencing. So let's examine, for instance, the most basic step, the first one. How do we identify bad channels? Here are some recommendations that I took from the websites of the tools that we are using. If you look at brainstorms, they say the original signals for a bad channel would be completely off or totally flat compared to the surrounding sensors, but also some bad channels are more difficult to identify. Now, typically we would use visual inspection, and as Phil mentioned it, this is often a subjective decision, and this is why we often rely on statistical thresholding and other techniques like recommended in many Python, which is source space separation, ICA, and so on. In any cases, there will be too many parameters to track just to report this in the method section. So here is an example that I used to clean some data. While it would be obvious to identify many of these bad channels, I cannot specify in the method all these parameters. Here I use the EEG lab pop clean raw data function. And as you can see, there is a bunch of parameters. Some of them indicate that the channel shows no signal for more than 5 seconds, it's less correlated than others, it has a line noise above 4 standard deviation, it includes a high-pass filter, and then I use what's called artifact subspace rejections, and finally clean up the windows. Only by sharing such a script, we can reproduce the analysis. So in this course, The different lectures will be given on different aspects of such a pipeline. Dr. Lange will discuss modern digital filters. Professor Makeig will present IC label, which is a machine learning method to identify artifacts from ICA. Professor Yao will introduce REST, a new method for zero-line reference. Dr. Gramfort will discuss normalization techniques, which are necessary before source modeling and multivariate analysis. And finally, Professor Puce will discuss modern EEG acquisition systems. These allows recording in the wild, but also come with new sets of pre-processing challenges. Just to give you an idea of the type of information you can get from these lectures. If we look at filtering, for instance, here is a standard experiment with standard stimuli and the deviant. And if we focus on the panel E right here, we can see the differences in the P300 using 3 different filters. Importantly, we can see that some filters will affect only the deviant and not the standard. Again, if we don't report properly what type of filter and the parameters of these filters, it would be impossible to reproduce the analysis. Now, what's interesting is that you will see in these lectures that filtering also affects ICA. As many of you know, ICA produce the maximally temporally independent signals available in the channel data by unmixing distinct and independent source contributions. But again, in ICA, many algorithms are available, and these will give slightly different results. From ICA, we get different sources. Some of them will reflect artifacts, such as muscle activity, eye movements, and so on. And typically, this requires expert knowledge to identify those. Now, what Professor Makeig will show in his lecture is IC label, which is a machine learning technique in which we incorporate expert knowledge on using training datasets, and then new data, new components can be classified automatically. Once the data are cleaned, we typically use re-referencing, which is again an apparently very basic practice, often done by subtracting the values of a channel from the average reference, also known as CAR, common average reference. But there's actually more than 10 different approaches. And the basic procedure influences the channel statistics and the clustering because the amplitudes change locally. Professor Yao will show how modern Bayesian methods, such as the REST, reference electrode standardization technique, alleviate many of the assumptions of average reference and other techniques. Finally, modern EEG includes multivariate analysis and source reconstruction, methods that depend greatly on data normalization. Dr. Gramfort will show that the choice of method changes the signal and therefore affect the replication of statistical findings. For this course, we created a GitHub repository in which we will access some data and be able to reproduce some of the examples shown in the lectures. And to conclude, I will recommend everybody to go on the COBIDAS WordPress report and look at the reference therein, along with these 2 other articles. Thank you.
Hello, everyone, and my name is Dezhong from UESTC Chengdu, China. And now my topic is EEG re-referencing. Common average re-referencing should not be your default. Now, first, I will briefly introduce the background about the reference. And this paper, we can see that the publication of EEG is the #1 among the non-invasive tools, and only the 2009 to 2016, EEG was overwhelmed by fMRI, and now fMRI has gone down. That means the EEG will have a rate lessons. This figure, we can see that the procedure of an EEG study, first we collect the EEG data and analyze the data and then infer in the brain. The reference is an absolutely fundamental aspect of EEG ERP recordings. If you don't fully understand the reference, you won't understand the signal that you are recording. And now I will briefly introduce the history of EEG reference. Human EEG was found in 1925 and formally reported in 1929, and from 1929 to 1968, Berger punched 14 primary papers. He examined bipolar and unipolar referential recordings, and the reference effect was known at the beginning. In 1960s, the Gibbs and Lamps team studied the EEG patterns of seizures. They failed to localize the orange of the psychomotor seizure because the reference electrode linked to the air's distort temporal activity. Using this air reference, most of their localized dysrhythmia were found in the frontal and occipital rings. The average reference was proposed in 1950s, and for dipoles in a cipher, the integral of potential over the surface is 0, and this is the key papers about the reference average reference. In 2001, a new method to standardize a reference of a scalp EEG recordings to a point and infinite, named the zero reference, was proposed by Professor Yao. Now these figures, we can see that the waves and the patterns are different for different reference, and the different we can see that the different referencing singles, such as the average reference single. This is a reference singles over average reference. This is a referencing singles over CZ. We can see that this is almost the non-zero referencing methods, and the rest singles is almost the 0. The lost single in the unipolar reference recordings and in these papers, the luck says that the first things I look for in a method section are the feature settings and the locations of a reference electrodes. Until you know how the data have been featured and referenced, you can't know how to interpret the ERP waves. The ERP wave has the active site, so it's hard to evaluate an ERP wave if you don't know which sites were used to create the reference portion of the single. In 1994, the IFC guidelines for topographic and the frequency analysis of EEG and the ERP was proposed. In this guidelines, EEG data, they say that the EEG data should be examined in several different multities, and the references should include the possibility of linked airs, linked metabole, nose, chin, neck, or other non-cephalic single size reference. The choice of which reference to use should be made after an initial inspection of the data on bipolar polygraph display, identifying active areas and choosing references that are relatively inactive. And in these guidelines, there is also no standard or no recommendations about the reference method. And in 2000, there are another guidelines for using human event-related potentials to study cognitive. And in this guidelines, it mentions that the experimenter must specify the reference. A variety of reference can be used depending on the type of ERP. When comparing forms to literature, it is essential to consider difference in the reference, and it is often helpful when comparing waveforms with those in the literature that use another reference to plot the waveforms using both references. Also, in that time, there is no standards or no recommendations about the reference method. And now, next, I will briefly introduce the reference program in a sense of algebra approach. Suppose there are n electrodes, we actually have n-1 channel recordings yi, and I equals from 1 to n plus 1. They are potential difference between active and the reference electrodes, and we can get these equations. And what we need are potentials at xi, the n recordings with 0 reference. The problem is that we have only n-1 equations, but our loads are n. It is an undetermined problems and needs supplementary equations. For special case 1, if we know that there is an electrode where the potential is 0, that means the xn equals 0, then we actually have n equations. then the equation set is solvable. The practical online unipolar reference assumption is that the nodes must achieve a CZ where the xn is 0. For special case 2, if we know that the sum of all channels is 0, then we actually have n equations, and the equation set is also solvable, and we can find xi. The practical average reference assumptions is average of the all channels over the scope surface is 0. In special case 3, if we know that the sum of the two air is 0, then we also have n equations. That means we can get additional equations x left plus x right is 0. Then the equation set is solvable, and we can find xi. The next airs or Masteur's assumption is that average of the two airs potential is 0. For special case 4, based on the fact that the potentials difference recordings by yi are produced by sources S inside the brain, and after we get the xn, we get the other xi from the known yi. The rest zero reference approach is that the supplementary equation is based on the fact the potential is generated by sources S. So I will give a mini summary about the reference problems in instances of algebra. In general, the scope reference problem is an undetermined problem. It doesn't need a supplementary equation. The supplementary equation for each of classes for cases 1 to 3, the online unipolar reference AR, LM, and so on, is assumption. The challenge is that these assumptions are unreasonable in general. Who knows where the potential is 0? Why should the average over the scope be 0? Why should the average of the two airs be 0? For the rest, the supplementary equation is based on the fact that the scope potential is produced by sources inside the brain. It may be reasonable. Next, I will briefly introduce the reference problem in a sense of physical approach. This figure, we can see that the dynamic brain activities in the volume conductors, and the white line here shows the point which with the potential is 0. We can see that these lines are changing time by time, and the low point where the the potential is constant. For the physics behind the average reference, the AR, the zero assumptions is the surface potential integral is 0. For this water metals, we can see that the surface potential integral of a spherical head is 0. But the surface potential integral of a hemispherical head model is not 0. And this was proposed in Yao's paper in 2016. And for average reference, we can know that the relative errors of average reference should be affected by the electrode density, that is the number of channels, and the coverage, and also the head shape. In this paper, the next says that if the studies use the average of all sets as the reference, you should check to see whether the electrodes are broadly and evenly distributed over the scope. If not, then the average reference may be inappropriate. And if the manuscript uses the average reference and doesn't provide a list of all the electrode size, then that's a problem. If you wish to use the average reference, you should use a large number of evenly spaced electrodes that cover more than 50% of the surface of the head, and you absolutely must report all electrodes that contributed to the average reference in the method section of your papers. Now I will briefly introduce the rest technologies. And first, with the theoretical zero reference, we get the equations 1, the x equals gs. This is the general equations. And with an actual reference r, we we have the equations 2, y equals gr. R means the reference r and multiplied by the s. From equation 2, we have s equals the gr inverse multiplied by the y, and combining equation 1 and 3, we have x equals gs and equals to ty, and finally we get the equations for x equals to ty. That means source S is adopted as a bridge to link y and x. Actually, we do not need to know s, but to know g and gr, which are determined by the positions of sources and the electrons. The problem is that, also the question is that the inverse problem of source is very difficult. Now next, we know that the non-uniquenesses of EEG inverse, that means for potentials or recordings, EEG potentials or recordings, we can get the y equals to G1. G is the leader field and S is the source, and G1 and S1 also equals G2 multiplied by the S2. So that means that is the principle of equivalent sources, and that means the S1 and S2 are equivalent sources of each other in producing the same scope potential. And for the dipole layers on the cortical surface, maybe the equivalent source of arbitrary sources in the brain. And based on the equivalent dipole layers on the cortical surface, the G, GR inverse, and the T are known, and the equations 4 is reasonable. Now I will give a mini summary about this section. First, the assumptions of AR is unreasonable, and because of the spherical head and the whole surface recordings and the dense recordings. The assumptions of rest is reasonable. That means the potential is generated by sources inside the brain, and that is a physical fact. And the sources may be equivalent sources, and that is solvable. At last, the rest is more physical reasonable. And the next, I will show some simulation evaluations about the EEG referencing methods. And in this work, we can see that there are different variants of rest, and the relative errors of the different variants of rest is lower than the relative errors of the average reference. That is the average reference of the error, and in different scope to brain conductive ratio. And the figure B shows that for the different number of channels, the performance of rest is also better than the average reference. This means that the rest is not sensitive to the head model and the perturbed head model. And this figure shows the results that for the wider coverage channels, the performance of rest is also better than the average reference. We can see that the relative errors of the rest is very, very low, and better than the average reference. For the typical brain networks, in this paper, authors also tested the performance of rest and shows that the relative errors of rest is the smallest, and regardless of the number of electrodes and the single-to-noise rate. In this paper, and using the realistic head model with 256 channels, the relative errors of rest is much smaller than the average reference. We can see that this is in different brain regions, the relative error of rest is the smallest. And in this paper, also the authors compared the most commonly used EEG referencing technologies such as REST and AR and IR. In this paper, it shows that using the simulated data with a single source shows the relative errors of potentials due to the re-reference. We can see that the errors of rest is the smallest, and the rest is the best for almost for all cases. Next, I will show the real data reference effects. In current practices, in clinical linked airs, the ALM are common used in quantitative EEG, the QEEG, and in psychology, the ALM and the average reference are widely adopted. And this paper using resting state EEG recordings and calculate the power maps or in different frequency band, and with also the different reference, we can see that for the different reference bands, the power maps is different. And in alpha 1 band and alpha 2 band, the results of rest reference is more reasonable. This range was located in the occipital rings. That's very clear to the occipital channels, the O1, O2. And this figure shows that for the other band, the theta band, the beta band, beta 1 and beta 2 bands, the power maps or rest is more reasonable. That which in theta band, the location was the power was located in the partial rings, and the beta band also on the occipital partial rings, and also using you can see that using the different reference methods, the results are differently different. In this work, we can see that the coherence maps over EEG during the resting state and with eye closed and in alpha band, the rest better reflect the partial occipital interactions in alpha band. And for other referencing methods, the performance is not very good, and especially the FCZ reference and IM reference. Also, in another paper, the authors investigated the influence of the reference on experimental effects between two conditions. In this paper, the authors use a simultaneous audio view stim and N1 peak. And here we can see that using the different referencing methods and the ERPs at the pink of ERPs distributions are different, and compare two conditions, we can get the different results. And this brings on the scope is for using ALM reference, we can get the different waves are located in the partition and the frontal rings, and for using average method, we can get the similar results located in the frontal and the partition rings, and we also with some in occipital rings. And using the rest reference methods, we can get the results located in the occipital rings. Also, in this paper, use the narrator to locate the sources of the ERPs and get results. We can show that the different waves are located in the occipital rings, and we can see that the source of a different wave is at the occipital and support the results of rest. In this paper, authors investigated the latency of scope time varying networks. Using the different reference methods, you can get the different results, and the latency of AR is larger than the rest reference. And that means if the reference single is non-zero, the subtraction would destroy the amplitude. And if the reference single has a delayed phase compared with other active channels, it would affect the latency as well. Also, in this paper, authors compared the different EEG reference choice for ERPs extracted by the ICA, and the results show that there are non-negligible effects of different reference methods on both temporal ERPs and the spatial topographies of some independent components. And also, we can see that compared to CZLM, the waves and almost are very similar, but for the topographies using the different referencing methods, the distributions on the scope are different. And overall, compared to the CZLM and the AR, consider both the performances of temporal ERPs. The rest reference has overall superiority. There is another very interesting work about the global brain EEG single, and that is in this paper, the authors get the using the simultaneous EEG and fMRI recordings and get the global single of resting state fMRI and define the global single of EEG, that is EEG with rest minus EEG with average. Then the authors calculated the correlations between the global single of EEG and the global single of resting state fMRI. And this figure, the results show that during the eye open, the gamma band was positively correlated. The global single of EEG in gamma band was significant positively correlated with global single of fMRI, and alpha 2 band, the global single in alpha 2 band negatively correlated with the global single of resting state fMRI. And during the eye closed, the global single of EEG in gamma band positively correlated with global single of fMRI, and the global single of EEG in theta, alpha 1, alpha 2, and beta 1 negatively correlated with global single of resting state fMRI. That means the rest also has the physical background or physical. So I will give a summarize about the reference methods. First is the reference has distinct effect on the waveform, the related parameters, including amplitude, latency, coherence, network, spectra, symmetry, and covariance, statistic test, and so on. And the rest is the best one to approach to the ideal zero. And the rest has been adopted by more than 100 papers. And now rest has been integrated into EEG Lab and the field trip tools. And also is recommended by the IFCN guidelines or in 2019 and recommended by the OHBM. Now this is a briefly introduce the EEG usage. And for the off-night rest, and the conditions are the nice coverage and at least upper hemihead surface and have enough density. That means the number of channels larger than 16, and the head shape is approximately low, and you want to have a result more close to truth. And the best choice for most cognitive study and clinic EEGs may be the rest. Next, I will introduce the demo of EEG rest. And now, first I will introduce how to use the EEG plugin variants of rest. And to install rest, you can download the zip files from the rest website using this link, or download the toolbox from the EEG Lab plugin website, and then unzip the place and folder in the plugins folder of your existing EEG Lab installation. So something like the EEG Lab plugins and the REST reference and the EEG plugin REST .im exists. To run EEG, to run REST, ensure that the correct EEG Lab folder is in your current MATLAB path, and run EEG Lab as a command from the MATLAB command window, and then load data using the EEG Lab. You can click the REST and the re-reference into REST, as this figure shows. here I also we also provide an example data, and this example data is pre-processed EEG data with average reference and with 62 channels, and the sampling rate is 500 hertz. And this is the ERP data, and the task is a video audible P300 task, and contains 30 target trials, and the label is S22. And the more details about this example data you can find in this paper. This paper below and the scientific report by. And now we will show how to referencing your data to REST step by step. And first, you can select the original reference of the EEG data. And for this example data, default is average. And then select the channels you want to reference. Please, it is noting that do not select non-EEG channels, such as the EOG, EKG, and so on. Only select the EEG channels you want to reference. And for this example data, the EEG channels is from 1 to 31 and 33 to 62. And also there are options about the retain remaining channels. That means if you want to keep unselected channels in the reference data, you can check the box. And step 3, if the channel location has been imported in EEG channel locations, this variables, and press button run directory, or you can select the user-defined need field file, and then press the button run, and then you can, if you think the result is okay, and you can press the button OK to save the referencing data to workspace, and this data will be saved in the OHBM in EEG lab, and in EEG lab you can click the data sets and to save the reference the data and labeled by the REST. And the next, you can use the EEG lab to show the results and click the tools and extract approaches and plot the waves such as the CZ channels with REST reference. You can get the ERP wave with REST reference. And this figures shows that by generally and by visually inspecting the EEG figures of average reference and the REST reference, and the similar EEG waves were observed roughly. That means the singles of the REST and the average is very similar. But next, I will show how the difference there are difference between the two reference methods, and this figure shows that the P300 waves of average reference and REST reference for CZ are showed, and also the topographies of the AVG and REST reference are also displayed. We can see that the ERP's event-related potentials are different from between the REST and the REST and the average reference. The gray rings represent the significant larger single intensity of a P300 for REST than average reference. Also, the REST in field has been integrated in field trip, and you can visit the homepage of the field trip and download the field trip to use the REST. The usage of REST in field trip also is very easy, and this slide shows that the example manuscript about to represent the single to REST reference, and you can set the parameters like the reference methods and input as REST, and input the need field as the variables, the CFG point need field, and also you can set the channel reference channels, and the default is all, and use the functions of field trip, FT pre-processing to referencing your data to the REST reference. And also other information about the REST, including who use the REST and the applications about the REST, and all other information or manuals and the information how to use and so on are all available on the REST website, and also the REST is free. Okay, at last, I will thanks to the people who partly contribute to REST and its evaluations and applications in the past 10 more years. And I will thanks to the international Professor Pedro Verde Sousa and Fernando and Robert and Alan and Chen for cooperations and discussions about the REST. And the works are supported by the NSFC, MOST, and MOE. Also, I will thank the organizations for human mappings.
So hello from home here in Asheville, North Carolina. This is Scott Makeig talking about ICA, using ICA for artifact removal and the IC label application. The work I'll talk about, of course, hasn't been done all by me. We have a large lab the last 20 years, the Swartz Center at UCSD. Our most famous project is the EEG lab software used around the world. And this was an occasion where we pulled together people when we realized it was the 10th anniversary, but there are a lot of other people that weren't there that day, many more, who've contributed to the richness of the EEG lab software environment. And in particular, we'll talk about the plugin application by Luca Piontonacini, who graduated last year with a PhD in electrical and computer engineering, and did a very good job in coming up with the concept for and actually completing the neural network training of independent component classifier, which is now available through the EEG lab environment. So starting at the beginning, EEG has been around for a long time. As early as '57, Gray Walter in England built a machine that had analog light bulbs here, and they would be twinkling in time with the alpha rhythm, and they had them in topographic array. Apparently had a 22-channel array. So this was the first EEG scalp map. No computers, of course, then, or no personal computers to display it on. Here is the era of cognitive ERPs, where the focus returned to a single scalp channel analysis. By '97, we had begun building the software for EEG lab, making use of MATLAB and advances in computer speed. Now you could actually make movies of the actual scalp map and then study its dynamics. And currently, this is work with Zeynep Ekelen-Achar. We're looking at high-resolution source imaging of EEG. And then from there on to network interactions at high speed, which I think is the current frontier of cognitive neuroscience. But I always like to start a talk like this with, bringing people back to the original question: what is the scalp EEG? After all, this is a sculptor whose sculptures are clouds that he makes hang in the gallery space. And I use it because for most people, even most people who have done EEG research over the 90 years, EEG is some sort of electrical activity that's somewhere in the head and so on. It's been very vague what it actually corresponds to in terms of the brain. We know that scalp EEG is a small portion of cortical electric activity. It doesn't all arrive at the scalp surface. And therefore, it's an even smaller portion of the total brain electrical activity. But which portion is it? How is it triggered? How is it modulated physiologically? And what is its functional significance, or what is the functional significance of those modulations? We know that EEG at the source level of the cortex is complex and not well studied. There are many influences on the cortex, within or on the cortex that are known to modulate the EEG rapidly in most cases. And there's a lot that hasn't been studied about those interactions. There's also a layer structure in cortex, of course, and the dipolar, the battery-like projections from cortex to the scalp are not actually of an infinitesimal equivalent dipole battery, but they have a layer structure. So there's all the complexity that really has still to be focused on by neurophysiologists. I always remember that brain dynamics are inherently multi-scale in a way that none of our devices are yet. And at each spatial recording scale, the signal, particularly electrical or electromagnetic signals that one records, each signal that one records, each channel that one records is actually produced by active partial coherence of distributed activities at the next smaller spatial scale. So if you recorded somehow from the bouton of a neuron, you'd get the partial coherence in time and space of molecular events, synaptic events. If you record from inside a cell, you're seeing the results of partial coherence in time and space of thousands of synaptic influences and non-synaptic influences as well, so-called ephaptic influences. If you record outside a cell, you're again seeing the coherence in time and space of neuronal currents associated with this neighboring cell structure. And if you record farther away on the surface of the cortex, again, you're looking at synchronies in time and space. Net, after subtracting all the discoherent activities, positive plus negative equals 0, you're seeing the spatially coherent activity that net is volume conducted to the scalp surface, where an EEG electrode can record. Now, an important fact that has been discovered or rediscovered in recent years is that information flow in this process is bidirectional. That is to say, it's not all signs of what's going on at the potential, the roar of the crowd coming up to the electrode. Actually, of course, the scalp electrode has nothing to do with the brain. And then what goes on in the scalp stays on the scalp. It doesn't influence the brain. But what goes on, what can be recorded at the cortical surface, in other words, the extracellular potentials, actually have been shown to affect what goes on inside cells. In particular, in their spike timings, which put together strongly affect information flow and even plasticity in the brain, learning. So there's a complex interrelationship and dynamic interrelationship between electrical potentials and the electrochemical interactions that they index at different spatial scales. That makes the brain particularly interesting and difficult to model. So let's now talk about the concept of an EEG source. What is the source of the EEG? Well, there's many sources. What do we mean by an EEG source? Here's a picture of the sun taken by this little telescope. NASA wanted to know how the guy got such a great picture with his little telescope. So have we seen something like that for the cortex, and particularly the human cortex? And the answer is no. This probably has 1 million pixels, this image. No one yet has a 1 million channel recording of cortex. And of course, the cortex is convoluted. You can't go inside all the sulci. So it's a very difficult problem. And there, until now, spatial temporal dynamics in the brain have not been the focus of basic neuroscience, who for decades, generations, have lived on trying to model the brain in terms of the spike histograms or the spike rasters from 1 and now maybe as many as 250 cells. But these multi-scale spatial dynamics have not been studied. So that's the situation we're in now. If you look at very low spatial resolution recordings, for instance, the clinical ECOG recordings where the electrodes are on the brain surface, you see something very complicated. There's obviously spatial temporal complexities here. It's obviously of very low spatial resolution. At any spatial scale smaller than this, we're also going to see complexities, as in the sun. It was Walter Freeman, the pioneer who had an MD and an engineering degree, who applied small grids to the smooth cortex of animals, rabbits, cats, and looking through an 8x8 grid about the size of my little fingernail, he saw the turgid pattern of phase, discoherent phase at 40 hertz was the frequency he was most interested in, suddenly changed to a smooth phase portrait that looked in his images like water drops hitting a pond and then rippling outward. Sometimes it rippled inward. He called these phase cones. And they occurred at particular moments relevant to the cognition of the animal. So on olfactory bulb, these would occur when the animal sniffed a new odor, not one of the regular odors he was presenting, but a new odor. So he realized that this phase coherence could have important and profound effects in terms of information flow through the brain, resulting because of the coordination of spike timings for distant activity and the relearning of local connectivity in the olfactory bulb itself. Now, if people who are doing models of thalamocortical coupling, this is a fairly simple model by Stan Anderson at Johns Hopkins. You can see this sort of pond ripple phenomenon easily and how it occurs biophysically. Now let's turn to the EEG. Here is an electrical head model put together by Zeynep, Kaeland, and Char. And I asked her to show a synchronous activity across some small portion of cortex. Here, projecting blue, which is our code for negative signal. And on the right, we see, and the biophysical fact is that the signature on the scalp of such a projection from a small patch will be equivalent to projection from a very tiny or infinitesimal battery located in the middle of the patch. If you had such a, this is just a model, but the equivalent dipole would have the same scalp map. But here she can actually project to the surface of the cortex and to the surface of the scalp and sum up all those voxels on the cortical surface that she has colored blue there. And when she does, she sees that that small patch of cortex actually projects to all the brain. To all the scalp surface, except for an equator line here where the potential is not registered. Now, in an actual EEG recording, there's always two ends of the channel. So for instance, if you had this channel somehow, that would be called F4 to P4, maybe something like that. It would record no signal. Why? Because both of those electrodes receive the same signal from this source. So that channel would be invisible to this source. Well, what sources does an EEG channel see? So here, Zeynep has created using an electrical head model for a subject, has created a channel here. We might say that's what, F8 to A2 in 1020 language, and then went to the subject's head model and said, well, what is that channel sensitive to? It turns out it's sensitive to everything in this picture that's not bright green. So each EEG channel is a spatial filter, but it's a very messy spatial filter, has not dominated by a single cortical area. In fact, if you want to look in terms of the math of it, this channel AB is not represent just the recording from channel A. That's not the correct model. Even if B is your reference like electrode, your channel AB is not the same as a channel sitting right on site A. In fact, it represents the difference between all the currents flowing to both of the electrodes, one in a positive and one in a negative. So here is again the receptive field of that channel. And to quantify it at a given instant t, the voltage in that channel, voltage on AB, is the sum of all the voxels in cortex times all the voxel activities in cortex times this map that we see here, this map of projection strengths. So VR are all the radial currents exiting, the net currents exiting the cortex at each cortical element, each cortical voxel in your model, or each cortical pixel in the surface mesh. And you multiply that times the weights that are shown here in colored, rather either positive, 0, or negative weights. So it's the sum over all voxels of the voltage emanating from that voxel times this complex filter function. That gives you the voltage at time t. Now take another time. All the activities have changed in all these colored parts of the WAB filter, and so in the parts that are shown colored in the WAB filter, that means the voltage at that time is going to be a sum and effective sum and difference of all sorts of different activities. So what's recorded from this one scalp channel is not one area or one brain process. It's a complex gamish of local cortical activities. So EEG is taken as a scalp channel signal is a very fuzzy brain. It's hardly worthy of the name of brain imaging. Each EEG records variations in a double-ended voltage difference between two electrodes, or two sets of electrodes. Each EEG channel thereby constitutes a particular spatial filter, receptive to sources located all over the brain surface, but particularly to a complex distribution of cortical areas and not to one radially oriented bit of cortex located directly below one of the two electrodes. So I asked Zeynep to make an animation. I said, well, take your cortex and separate it into 1 million voxels, each the size of a mini column, and give them all in EEG waveforms, but independent waveforms. And so they're all doing their own thing, and there's no synchrony. and then see what happens to the scalp EEG signals that emanate from this very busy brain going about doing EEG. And so that occurs at the beginning of this animation. And then I said, well, don't change these signals except to make some of them phase coherent. So they're varying coherently instead of discoherently. And don't change the amplitude, just change the degree of spatial coherence in one patch of cortex. And then we'll see the effect on the scalp EEG. And if we're lucky, this will work. So here's where the patch of coherence will emerge. And here is the projection screen, the scalp. And so this will represent the degree of coherence in that patch. So we see now there's almost no EEG. Everything is phase canceling itself out. All those million sources. Now the sources are just as active as ever, but now there's large EEG because of the spatial coherence, where those sources are not canceling each other out, positive and negative. So this patch, this locally coherent patch of voltage changes, potential changes, is in fact an effective source of EEG. It's a dominant effective source in EEG, whereas all the activity that's going on in the brain, the other 990,000 mini columns, because although they're very active, because they're spatially incoherent, they make very little net effect on the brain, on the scalp signals. So effective sources of the scalp EEG are, I would argue, emergent islands of cortical local field potential, synchrony or near synchrony. So so how does ZANEP make these models? Of course, you get an MR image or more than one if you're thorough. You segment different types of brain tissues that have different conductivity profiles. And you use the head geometry from an MR image to build boundary element or finite element models of the different head tissues, such as this, and her NFT toolbox will do that. And then given EEG or MEG data with careful co-localization of the sensors to the head model, and appropriate signal processing, you can come up with an inverse problem. An inverse problem is a map or a set of maps of scalp activity for which you want to ask the question, what source activities, cortical source activities are producing these? And if that map is a simple map, unlike almost any raw EEG map that you could pull out of an hour of recording, if it's a simple map that represents the projection of a single area, such as the effective source we saw in the last animation, then it is possible to solve the inverse problem and estimate with much less uncertainty than otherwise the source of the spatial coherent patch, that is the patch in which spatial coherence emerges and produces a large EEG signal. So how to find those simple maps? And that was 25 years ago that I came across a paper by Tony Bell, Terry Sinofsky, on the cocktail party problem. You have multiple sound sources and multiple microphones, each one's recording cocktail party noise. And it was some French engineers who thought, well, it should be possible from these multiple views of that, recordings of that noise, to find the individual sound sources simply by the fact that they are temporally independent of each other. That is to say, knowing the sound pressure level at time t in the man's in front of the man's mouth is going to tell you nothing about at that instant the sound pressure level coming from the man's saxophone. Excuse me. So that was the independent component analysis. And Tony Bell developed an elegant algorithm. It turns out to be nearly the best for EEG analysis still. And I had the idea when I read the paper one night that in preprint that this same situation occurs in EEG, where you have multiple sources and they project to multiple scalp electrodes. They sum linearly there. And perhaps with ICA, you could find the time courses of activity in the individual cortical sites and their projection patterns, their simple maps representing the projection of a single source. So a week later, we published or sent to NIPS the first paper on ICA for EEG and have been at it ever since with increasing success, I would say, and confidence. So are cortical or other sources of EEG signals actually temporally independent of each other? Well, for the cortex, the question is serious because, of course, the brain is interconnected and operates as a whole, ultimately, to organize our behavior, our thinking, our experience. So imagine some domains in which local cortical synchrony could erupt, becoming thereby, at a moment, efficient, effective sources of the scalp EEG. Well, the sustaining of potentials and this emergence of coherence comes from the coupling of excitatory and inhibitory cell networks. Inhibitory cells have no long-range processes. That means that this coupling has to spread locally. So it has to spread through a domain, just as Walter Freeman saw in his pond ripple picture or his phase cone picture. The coupling of cortical areas other than local, other than neighboring neurons, coupling is vastly sparser, 1 million times sparser. So that these areas have much more dynamic interconnection with themselves than any other cortical area, typically. And therefore, and furthermore, the thalamic-cortical coupling, which also organizes and can sustain and helps sustain cortical rhythms, has very narrow pencil-like coupling. Those two facts alone mean that the concept of relative independence of temporal signatures or of time courses of activity in different effective sources is physiologically highly plausible. And so the concept of temporally independent domains of local synchrony is physiologically plausible. Now, of course, and the scalp projection of such an effective source will be the equivalent to that of an equivalent dipole model. Now, of course, some cortical patches are coupled by white matter tracks, corpus callosum being, of course, a prime example. In that case, you might have synchronous activity in two patches whose joint scalp map would be the sum of two tiny model equivalent dipoles located in the center of the patches, the centers of the patches. So ICA solves the inverse problem by separating into two problems. First, what sources are active, and two, where are they located? So it gives a model-based response to the first questions. What are the effective source processes? And it gives their time courses, and it gives their independent or their individual projection patterns to the scalp. For brain sources, it also then helps to answer the second question. Where do these sources originate? Because you have the simple map that represents the projection of the source. So it helps, but it leaves you, ICA leaves you with an IC classification problem, which independent component process comes from the brain, which comes from eye activity, which comes from muscles, which comes from line noise, which comes from heart, and so on. Now, the beauty of using ICA and why we persevered all these years is that now you change from these very messy spatial filters that are scalp channels into a set of very coherent, effective source processes, which you can localize and which have much higher signal to noise than channel recordings. So in fact, when we look at clinical data, the amount of information about individual subjects contained in the source components is much larger than their mixture recorded as scalp channels. And it's quite amazing how closely these maps can resemble the projection of a single cortical patch. Or here, two bilateral patches, bisymmetric patches that could be easily coupled by corpus callosum. So, and you can look at many of them at the same time, and you can look at their interconnections and their in relationship to experience and to behavior. Here, for instance, is sources, brain sources retrieved from one subject playing a very complicated video game for an hour. When I looked in the fMRI literature for the functional correlates and fMRI experiments of all these places, it had to do with spatial memory. This was a world video game. There's multiple screens, multiple instructions. It had to do with manual control. It had to do with manual interconnection with space. It had to do with multitasking. It had, et cetera, et cetera. And just, in fact, what the subject, cognitive processes that the subject was fully engaged in this experiment. But now let's turn to the pre-processing question. ICA also gives, separates out, that is, the prominent non-brain processes that contribute to scalp recordings. Here is a signature, the scalp map signature, and you can see in the activity plot, what we call the ERP image, that this has to do with eye blinks that occur, or vertical eye movements. Whereas this component map and this activity profile tells us that this component process accounts for lateral projections by the eyes into the scalp EEG when the subject moves their eyes left and right. The eyes are standing batteries, so when you move your eyes, it projects, it adds a different voltage map to the scalp. Here is a postauricular muscle. It's shown a little high here on this picture. This was the first time we actually used an individual head image and co-registered. But you see, here's the EMG signature in the spectrum, and here is the record during the trial, during the experiment, that this muscle was tense for a while, then it became relaxed, then it became tense, and then it became relaxed again over half hour or an hour of recording. Probably the subject wasn't aware of it, but it does contribute to the EEG signal. Here is a ECG contributions. Mainly they project to the very lower part of the recording array. If the array was here, I think we let it extrapolate down the neck. But when you look at the single traces, you can easily see that, as we'll see. And then here are scalp neck muscles. Their signature is a polarity reversal at the end of the muscle and the insertion into the tendon. And you can see from the location and from the angle of the polarity reversal, get some idea here in Gray's Anatomy what muscles they may be. I think someone is doing a PhD dissertation using needles to verify that this recording was of this muscle and so on. Well, I don't know which correspond here. So ICA separates out all these EMG contributions from distinct sources, just the way it does effective sources that come from the cortex. So here's an ICA example. Here's 15 seconds out of a half hour of recording from 100 channels. We show 9 channels here. And then here are 9 of the 100 independent component processes that ICA decomposition transforms the data into. And we see what we mean by temporal independence. You don't see any of this in this. You don't see any of this in this. You don't see any of this in this. And these are all separated simply by spatial filters. Spatial filters that cancel out activity from the sources of all these from all these places and leaving activity from one place. Here is ECG, electrocardiogram, which affects all the channels to some extent. And if we look here at this spike, we can see it in the posterior channels. We can't see it in the frontal channels here at all, but ICA separates it out very cleanly and neatly, because it creates an activity profile that contributes independent information to the overall scalp EEG. And on the basis of that, ICA can separate out. It separates out distinct sources of information in the data. Here is this scalp muscle, again, a post-auricular muscle with muscle spikes. Here is a subspace. Now the subjects seem to be blinking slowly after every trial. So rather than throw out the trials, you see the blinks are going all through the frontal channels of the data. But here ICA separates it out into an earlier and later phase of the blink and completely removes it from all the other source activities. Here is a frontal midline theta during a stretch of good performance of the subject who is doing a difficult two-back task. And here is another theta outburst in right frontal cortex during portion of this. And here is a bilateral alpha activity. So the subject blinked after every trial, and then there was a waiting period of maybe 500 milliseconds before the next stimulus appeared. And in this lateral occipital or parietal area, bursts of alpha appeared. That's what I call alpha flooding. And in red, I show the ERP repeated for this channel. So the ERP would say, well, this is what happens. In this experiment with ICA, you can see exactly what happened in every trial. So you can look at trial to trial variability, and that's what we're working on to understand more about the brain's dynamic responses to experience and anticipation of in our everyday life. So here is the Henson and Wakeman data. Here is the face ERPs. These are all the channels. This is the P1 potential, and here is the N170 peak. I don't call those components, I call them peaks to distinguish from ICA component processes. So this is the famous N170 face potential. So what does ICA say for this subject's data? There are two sources, left and right, of the P1, predominantly. And there are two sources of the N170. You see the top one is mostly right and some left. The next one is mostly left and some right. And together, here's their localization at bioequivalent dipoles in inferior temporal cortex. And you see together, they constitute almost the entire N170. These two processes sum up to almost this map, except for the fact that there's also positive potentials going on, peaking a little later, around 200, but with very complex sources. So an ERP person told me, well, yeah, I don't look at P200s because it's too complicated. But it turns out, for some reason, and just by chance, I would say, the N170 has very easily dissociable source processes. Okay, so now you've done your ICA decomposition, and you pull up at the EEG lab a plot of the scalp maps of the different components, and what are all these links? So are they eyes, muscle, heart, channel noise, brain? Well, over the few years we were putting together EEG lab, and before that we came to have a kind of a expertise in this, so that we can just look at the scalp map and say, oh, I bet that's eyes, eye blinks. I bet that's lateral saccades. I bet those are muscles. I bet that's a heart, the ECG. I bet these are single channel noise, and then these look like sources of brain activity and some others in here. Well, okay. What I forgot was, or what we forgot was that we had years to develop that ability, not only by looking at the scalp maps, but also looking at the activity profiles, the spectra, and so on. But no one else has years to develop that skill. So I was very happy when Luca showed up in the lab and said he would like to work on an IC classifier, and call it IC label as the project. And he had a unique way coming from machine learning to do it. He gathered all the independent components from all our data sets on over 20 years in the lab. They ended up to be over 100,000 ICs, independent components. And he made for each of them their scalp maps, their power spectra, their autocorrelation spectra, or autocorrelation kernels, and other measures. And so he got a huge database, but they weren't labeled as to what kind of components they were. So he began by trying fancy types of neural networks that use labeled and unlabeled data. He tried opponent networks, where one tries to generate fake maps and fool the other one who's trying to classify both the labeled and the unlabeled maps, etc. That should work if you have big enough data, but in this case, it didn't make any improvement. It was much more complicated. So he ended up using just labeled data. But where to get the labels? So the IC label website. And currently there have been 51,200 label contributions by not only Luca and me and so on, but by dozens of other people, if not hundreds by now. And it's open for more. This algorithm can be trained over and over again, as when this becomes 60,000 and 70,000 and 100,000 and 200,000 and 300,000 labels, it cannot but become more accurate. So what is the IC label website? Well, in the first place, Now, once you've trained the network, then making a MATLAB function or a Python function to implement the network by computing the scalp map, the power spectrum, the autocorrelation for a given IC process, and putting it through the network and coming out with an output. It'll come out with an output vector representing the probabilities, roughly, of that this source process is a brain, an eye. It comes from the eyes, it comes from the muscles, it comes from the line noise, it comes from channel noise or other. So when you go to the labeling website, and I would urge you to start with the tutorial, go right here, because there's a tutorial on how one learns to classify ICs based on all these properties. So he's computed all these properties and made pictures of them for 100,000 components. Here's the scalp map. Here's the data variance accounted for. This is a large component it makes a large contribution to the data. You see there's lots of channels, but it counts for 3.6 of the data. It's the third ranked by variance of all the ICs. Here is a raster plot. Colored lines are single trial ERPs. It's epic data. Here is a snapshot of some of its time course. Here is its spectra in two ranges, 3 to 40 and then 3 to 80. Here is equivalent dipole for this And you see it properly located in left occipital cortex, I believe. That's still occipital there. Or if you solve to have two dipoles, then it's located in mostly in the left, a little bit in the right. In fact, the ratio there is 7 to 1. So this is 7 times stronger than that. So whether this is makes a real contribution to the source, or is this enough? I don't know. The residual variance, that is to say, if you project this dipole through a head model to the scalp, the projection map looks almost like this. The residual variance from this IC map is only 3%. Now, then it gives you a panel here. So you press what you think this is. I would say this is a brain component. Look, it has an ERP. It has reasonable activity, although a little dominated here by a certain frequency. The scalp map is highly dipolar. So I would say brain, and then I would hit submit. Then there are 3 phases of the tutorial. One is that it will give you explained examples, where it shows one of these pictures and say, this is why experts select this as being a brain component or a muscle component and so on. So you can go through that tutorial. And then after that, it gives you examples with feedback. So that would be an example like this. If you give this and you see these in the time course, you realize these are blinks. So this is an eye. This accounts for activity from the eyes. Frankly, I don't know why this blue channel here, it may be actually mislocalized, because normally we would not see that. It would be bilateral. And then when you say, okay, I'm going to say I, or whatever it is you decide to say, and you can always add a question mark, or you can give more than one answer, then it will pop up this record of what some experts, that means probably me and Luca and I don't know, Makoto or Arno, said it was, and what 3 other users said it was, and they all said it was I. So you can then look again at it and see whether why they said it was eyes and why you think it's something else, and continue your learning process that way. So there's the explained examples, then there's the examples with feedback, and then if you choose to go on, you can register and just start labeling components without feedback, contributing new labels to make the algorithm work better. And to run IC label in EEG lab on a data set, you go to the tools menu, classify components with IC label, and choose label components. The default version is best. And then after it works a little while, and it's really quite quick, it asks you how many it should plot. It wants to show you the properties and the classifications of the components. And so you have some options here. You say, okay. And what you'll see is a map like this. Now in this map, you see these buttons. These buttons, if you click one, it will pop up another window with all those properties, or most of those properties for that component. It'll also tell you what the probability that it computed that component is an eye component, which would be very low, or a muscle component, again, very low, etc. And it gives you the leading component, the most probable classification here on the front page. 99.5% probability, this is a brain component. These aren't actually probabilities, but the sum is normalized to 1, so they can be considered to be probabilities. And here's 2, for instance. Here, this says it's brain, but only with 71% probability. So maybe you should click on the button there and investigate that further. Here's an eye component, eye blink. 94.4% probability it computes. Now this is, again, it's a stochastic estimate. Doesn't mean this is ground truth. So you can, again, look at its properties to see that you agree if you want. Now back to the Henson-Wakeman data. Here is a wonderful decomposition from one subject's data. And these are the first 32 components or 35 components. And we've added a little information here for a paper that we're putting out. These data were taken in the MEG shielded room, which is also an electrically shielded room. So the data are really quite beautiful. And as is the ICA decomposition here. So here is a muscle, here is a muscle, and you see the IC label now is given that judgment as well. Here's a muscle, here's a muscle. So all the individual scalp muscles are active. Even when the subject is reasonably quiet, he's still holding his head up, and that means that he's using all the neck muscles as needed. And those are contributing EMG activity to the EEG recording. ICA is separating all those out from the brain processes. Here's a brain process. IC label says probability 99.6. Here's another one, probability 100.0. Here's another one, 99.9, and so on and so forth. Here's a pair of them that are independent of each other, but they seem to be have some connection, right? So this is a subspace, and so on. So and here again is a bilateral occipital component process. So this can be looked at as pre-processing. I would look at this now as pre-processing of the data. You put it into original source space. You pay no attention unless you're doing a project on neck muscle tension and its relation to anxiety and depression. You might want to look only at the neck muscle or the head muscle component processes. But if you're interested in what's going on in the brain, then you can look at the brain processes. And need not worry that the brain processes are contaminated by muscle activity, or at least not in a major way. And you have many brain processes now you can look at independently or look at their transient couplings and decouplings according to the cognitive circumstances or behavioral circumstances. And you see so many processes you can look at. So this is only, IC label is the 7th or the 6th, well, 1, 2, 3, 4, 5, 6 attempt at creating a plugin for EEG lab that classified components. It's the one that classifies the most different types of components. And it's also 10 times faster than IC Mark, which was also machine learning based algorithm, came out of Lars Kai Hansen's lab at Danish Technical University. But it requires 10 times as much time to compute, and it doesn't have quite the range of specificity of IC label. So IC label, I think, is something I can recommend. If you have very large data, if you're doing 1,000 subjects and you don't have any time to spend on individual data sets, then you can learn to trust IC level to that extent. Although again, it's stochastic. It doesn't make it's not that it makes no mistakes. So I urge you to carefully look at results of IC label for many data sets, several to many data sets first. And there's a paper in Neuroimage where you can read all about the details. And please contribute. Please use the tutorial. It's, I think, a very valuable tutorial. And then continue and contribute some labels. You will, if you contribute enough, about 100 or so, then it will begin to rate you. Why? Because we don't want to have people who are pushing random buttons contribute to the trashing of the algorithm, basically. we want to pay more attention to people whose labels agree with the experts. So in the labels you're going to contribute, some will be of components that have been rated by experts, and that's basically to test you and to give extra information about those components. And then some will be new that have never been tested before or not by our experts. So there's a sophisticated scheme for choosing which components to give you. And you can contribute labels, and IC label will continue to get better for you and for everyone. So this is I think, a beginning of an exciting era in neuroelectromagnetic brain dynamics, and there's lots to do. And I thank you, and thank to Cyril and Aina for putting together this course. And I wish you the best in this pandemic time and beyond.
Hello, everybody, to the OHBM educational course about EEG data acquisition and pre-processing. My name is Nicholas Langer, and I'm an assistant professor at the University of Zurich in Switzerland. Today, I will talk about the effects of data filtering on automated ICA classifiers, ERP, and multivariate analysis. To say it right at the beginning, the topic of filters and filtering is quite complex. But don't be afraid and scream as the top plot of Philip here, which resembles to the art piece of Edward Munch. I will guide you smoothly through this lecture and provide you the most important information and give you recommendations to include in your daily analysis. So here you see the overview of the lecture. First, we actually need filters, and then I will introduce different terminology of filters. Afterwards, I will show the effects of low-pass and high-pass filters. Afterwards, I will present some problems when combining baseline correction with high-pass filters. And then I will show you some caveats when using filters with automated ICA classifiers. And I will provide you some information and problems when using high-pass filters in multivariate analysis. And finally, I will introduce standardized pre-processing toolbox, which is called Makeig. So stay until the end, so the best comes at the end. But throughout the talk, I will always offer interim summaries and recommendations for each of the topic. So let's start right away. Why do we need filters in the first place? EEG is quite a noisy signal, and different types of artifacts contaminating the EEG signal, which might be non-physiological, such as bad electrode contact or power line noise, but also physiological artifacts, such as sweating, eye blinks, or heartbeat. So all EEG analysis requires some kind of data processing. And data filtering is always applied in the process of EEG pre-processing. And the goal of filtering is always to optimize the signal ratio, which means to reduce the noise and maintain the actual signal. However, the filtering can produce unintended adverse filter effects and artifacts, which I will show you along the lecture. So the best way, we should already take some measures to improve the signal to noise ratio during the EEG acquisition. And we can do this by using proper electrode preparation to lower impedances, to comfort the patient while the muscle are sweating, and to prevent electromagnetic interferences by using some shielding, and definitely record sufficient number of trials. So let's start easy and look at the different types of filters. We distinguish between low-pass filters, which allow frequencies pass below a specific threshold and reject the frequencies above the threshold. And then we have high-pass filters, which is the low-pass filters. The high-pass filters above the specific threshold, all frequencies below this threshold. And then we have band-pass filters, low-pass and high-pass filters, and we have notch filter or stop band filters, which attenuates specific frequency bands. In EEG, band-stop filters are almost exclusively used to suppress power line noise, which is in the 50 or 60 hertz, depending where you live. And you, in the talk, I will show filters and notch filters should be actually substituted by newer methods. So let's look how an ideal filter would work like. So, we see here this blue line, and this shows you the cutoffs of the frequency. So we distinguish between the pass band and the stop band. The pass band are the frequencies that are retained, which keep the amplitude, and the stop band are the frequencies which are attenuated, which ideally have the amplitude of zeros. But those ideal rectangular windows, they do not exist because the world is less than ideal and applies to the filters. And this is due to the Fourier series, which says that any abrupt transition in one frequency or temporal requires an infinite number of components of the domain. So if we would want to have such a perfect rectangular filter, we would need the filters to be of infinite duration. But this is not possible because we have a finite number of filter coefficients. Thus, consequentially, we will get ripples, or called ringing artifacts or Gibbs effects. So ripples are defined as deviations from the expected frequency response, which you see here in green. And we distinguish between passband ripples, which is here in the passband, and the stop band ripples, which are here in the stop band. The main issue in our EEG analysis are passband ripples. So even in the frequency bands, which we analyze and provide our EEG analysis later on, there are some deviations in there. And no matter whether you filter on continuous data or on epoch data, you're always going to have those deviations from the signal in there. You should always filter on continuous data. So let's look at some filter parameters. As I said, those ideal rectangular windows, they do not exist. So thus, researchers different types of windows, and they distinguish each other between the max band stop attenuation, which means how well the filter rejects or dampens the unwanted frequencies. And they have also different passband ripples, which means how strong the distortions are in the frequencies which we actually retain. And they also differ between the transition bandwidth. So far, I showed you what passband and stop band was, but there is also this blue area between the passband and the stop band, which is called the transition band. And the width of this transition is called the transition band width, which is a window type, which I just showed you, and the filter order. The filter order defines the steepness of this transition band. And what sometimes also is used is the slope of the magnitude response in this transition band, which is termed the roll-off. So narrow transition bands, they have steep roll-off, and wide transition bands would have shallower roll-off. What is important to remember is that steep filters produce stronger signal distortions, which means wider temporal smearing and ringing artifacts. And finally, an important filter parameter is the cutoff frequency, which is here indicated by the number 1. And the cutoff frequency is actually the value which you should always report in your paper, and it always lies in your transition band. And for FEAR filters, which are often used, it's in the center of the transition bandwidth. And it indicates the filter effect is at half amplitude, which you can see here. How are the effects of filters actually measured? We use different filter responses. And what I just showed you is responses in the frequency domain, which is also called the magnitude response. Magnitude response, we can identify the filter characteristics, such transition bandwidth, how strong the distortion is in the pass band, how well the signal is attenuated in the stop band, and so on. But there is also a response domain, which is called the impulse response. And these filters are often tested with the very transient sharp signal, which is indicated here in the dashed line. And what you see here in the green, this is the output signal after this black dashed line is filtered. And what we can observe are two things. First of all, the filter creates a delay, but you don't need to worry about this because all of the filters which implemented in the toolboxes can correct this delay with no problem. However, what you should be worried about is that the filter actually smears the signal. So here we have a very sharp transient signal, but here the output is smeared. And we also see here some ripples, which you can also nicely see here in the blue figure. So this means that low pass and high pass filters smeared the signal in the time domain, which to remember for later on. And there are the parameters which are not as important. For example, we have zero phase filters, which are usually non-causal, and we have minimum phase filters, which are usually causal. And because 99% of all EEG publications using a zero phase filters, I'm not going to go into the detail, but you can read them here along. And also, there are different filter types, such as finite impulse response filters or infinite impulse filters. And I'm also not going too much detail, as they do not differ too much if they are both zero-phased. However, Wittmann recommends to use FEAR filters for most of the EEG analysis. And FEAR filters are also implemented by default in most of the EEG toolboxes. So let's take a look at the different filters in the different EEG software. And in particularly, let's look at the default filter lab, which is a non-causal zero-phase Hamming window sync basic FEAR filter. And what's important, and you have to consider, is that if you use this default EEG lab function, is that you provide to the EEG lab function actually the passband edge and not the cutoff frequency. So the passband edge is the edge of the passband. And remember, the frequency filter loses half of the amplitude. If we use, for example, a high pass filter here with the 0.5 passband, that doesn't get cutoff should report in your paper. However, each lab provides the cutoff frequency in the command window of the console of MATLAB, and you should get the cutoff frequency from there, or you can put it with the heuristic, which is provided in the help function. But I would claim that probably 70% of all EEG lab publications or people who use the EEG lab default filters report probably the filters in the wrong way. So please don't do this mistake as well. And you can also design your own filters in EEG lab. And then let's have a short look at the other EEG lab toolboxes. All of them are using a zero phase filter, and most of them are using a FEAR filter. So if you're interested in this, you can take a look later on. So let's get to the interim summary. The first important point is that sharper filters produce signal distortions, which means wider temporal smearing and strongest ringing artifacts. Thus, filters with wider transition bands are preferable where possible. And this is actually applicable for low-pass filters. Consider the EEG signal, which has a 1/f spectrum, and that the higher frequencies or amplitudes, thus a wider transition band designed is not as such as a problem. However, for the high-pass filters, we need steep high-pass filters, which should be used for ERP and other EEG analysis, because remember the 1/f spectrum in the EEG signal, we have very high amplitude for the low frequencies. And we should actually substitute bandpass filters by separate steep high-pass filters, shallow low-pass filters, because bandpass filters use low and high-pass filters with the same filter design, and this is oftentimes not ideal. Moreover, notch filters can produce strong artifacts, as has been shown in the publication of Stephen Luck in 2000. And they should be replaced by more modern techniques such as time domain regression-based approaches, which in clean line, for example, for EEG lab, but also by Alexandre by his Zeppelin, which was just recently published. And finally, it's important to report all relevant parameters and also correctly, especially consider the issue with the default filter from EEG lab, which distinguishes between passband edge and the cutoff frequency. So just to say one word regarding Zeppelin, I heard really good things about Zeppelin as it combines the benefits from spectral and spatial filtering. However, Aina suggests that oftentimes three components might work to remove all the power line noise. The Buckyman and Hansen data set and remove the line noise with three. However, there was still some residual power line noise included. But if I used 5 components and 7 components, it looked quite good. And this was also our experience for our data. However, we're using an eye tracker while we record the EEG data, and this might induce a little bit of power noise. However, I would always recommend to do a sanity check, which is also easily available and provides sanity check figures where you have on the left, you have the original data, and on the right, you have the cleaned data, but also the removed signal. And you nicely see that the power line is nicely removed here with 7 components. But what I'm also a little bit worried about is that also some frequency in the frequency range which you might also be potentially interested. And my PhD student, Marius Strengler, did the short analysis where he compared the power between the power in the frequency bands between the notch filters and the Zeppelin. He didn't find any difference in alpha and beta, but he found significant differences in the delta and theta band. So one should be cautious, and some further analysis should be conducted as well. So let's look at the effects of low pass and high pass filters in ERP analysis. I would here like to quote which says, "It is important to realize that changes in the frequency spectrum introduced by filtering must cause changes in the temporal signal, as both representations are coupled by the Fourier transform." Which means what I showed you already before is that low pass and high pass smear the signal in the time domain, which can be seen here. And distortions in the time domain also occur if you filter on continuous data, which you should always filter your data on the continuous data on the epoch. But all the effects I'm going to show you right now is all based on filtering on the continuous data. And the distortions will be even much bigger if you're on epoch data. So let's start with the effects of low pass in ERP analysis. So there is a famous study which is very often cited, which is by von Ruhlen. He used simulated data where he used the data which show an ERP component 150 milliseconds after the stimulus was presented here in black. And then he applied a 30 hertz low filter. What you can observe here in red is that the filtered data, the ERP component is smeared back in time, and the onset of the ERP component seems to start already at 100 milliseconds. So his conclusion was that if low pass filters are applied, the onsets of the ERP will be interpreted. But however, the peak amplitudes and the peak latencies are okay to interpret. And in the same year, Rousselet actually also published a study. He investigated different low-pass filters seen here in different colors. And he used real EEG data and found out that the different low-pass filtering has almost no effects on the onset and the time course of large ERP effects. So here we see the different onsets with the different cutoff frequency, and they're all more or less the same. And also the amplitude doesn't change as long as you use a higher cutoff as 20 hertz, which is recommended anyway. This discrepancy is coming from. So the von Ruhlen simulated the data, and the simulated data used the function for simulating this ERP component. And these step functions are particularly prone to ringing artifacts. However, Russellet also mentioned if the signal-to-noise ratio might be actually a little bit lower, that the low-pass filter distortions might influence. However, the fact that the higher frequencies have low amplitudes, remember the 1/f power spectrum, this effect reduces the risk of serious distortions of the ERP form when using low-pass filters. So a quite simple situation is to apply shallow low-pass filters with frequency higher than 40 Hertz. And when we retain those frequencies, we also don't change the components of ERPs, which are have a short peak and a short latency components, such as the P1, for example. So now let's take a look at the effects of high-pass filtering on the ERP analysis, which is often used as a 0.1 Hertz or 1 Hertz filter. And the problem is much bigger because many ERP components, particularly the slow components, which slowly increase in the P300, the N400, or the P600s, are affected by high-pass filters. And this can be nicely shown here by the simulation. Here we have a simulation of a P600, an unfiltered one. And when we use a 2 Hertz high-pass filter, first of all, the amplitude is diminished, but what is even a bigger problem, that new peaks are introduced through the filtering. We call this also inverted spreading. And this has also been shown by simulations of Thunder and colleagues, where they used also a simulated ERP in the P600 with different amplitudes, and then different high-pass filters with different cutoff. And what they observed is that the cutoff frequency, the stronger are those new introduced peaks. And it seems that there is suddenly a new ERP component, although this is induced by the filtering. And this effect is also stronger the ERP amplitude is. Thus, the distortions depend on the cutoff of the high-pass filter and the amplitude of the EEG signal. Thunder et al. Also investigated this in real data by applying different filters seen here in different colors. And they used, for example, a task where the subject had to decide whether the sentence they just read is grammatically correct or incorrect. And here the ERP differences are plotted. And usually the P600 is sensitive to the grammatical correctness. And when you apply very high cutoffs for high-pass filters, such as 1 Hertz or 0.7 Hertz, which you see here in orange or in purple, we see two things. You see a reduced P600, but very some is that you induce new N400 differences, which is actually not really there. And this is because what I showed you before, those inverted spreading. And this has also an effect on your statistical analysis. If you use high-pass filters with cutoff frequency higher than 0.3 Hertz, it induces artificially significant effects in the N400, which is actually not the case if you use low cutoff frequencies, which is in red or blue. So this might lead to potential false conclusions by the research, namely that the experimental manipulation is impacting multiple components rather than just a single component. And you lose a low cutoff frequencies, such as 0.01 or 0.1 Hertz, high-pass filters have only a small effect on the results, as I showed you here in blue and red. So I also performed this analysis with the data which are provided here in the educational course with the Wackermann-Hansen data sets, and applied different high-pass filters. And if you use pass filters with the low cutoff, you see that the differences between the famous faces and images is actually the strongest at the N170, where you would expect it. But the problem is if you use two high-pass filters, such as 2 Hertz, for example, seen in red, then you introduce differences between the famous face and the scrambled images here at the P100, which there are actually no present, just induced by the high-pass filters. So now I want to talk about baseline correction, especially when using in combination with high-pass filtering. So what are the baseline correction? As I showed you before, high-pass filters spread the activity in time, and it means backwards in time and forward in time. And it can spread it potentially even into the baseline interval, which is then subtracted again from the actual activity you're interested in. And in general, the problem, even if you don't use high-pass filtering of baseline correction, is that two conditions might actually show different baseline intervals. And this because the subject is memorizing some responses or have some systematic preparatory potentials. And you always have to consider that the baseline corrected measures are different scope. And because we subtract the post-stimulus activity from the baseline interval. And this is problematic if you compare two conditions with baseline corrected data, because there you have the underlying assumption that there is no systematic difference in the baseline. You only actually properly compare the post-stimulus activity between the two conditions if you assume that the two baseline intervals are equal. But this is oftentimes probably not the case. And there is an extensive discussion between the Tanner and Luck group versus the Myers and Wiedmann, resulting in 4 papers going back and forth, which is really entertaining to read, but for me, it's difficult to take home message from this. So I claim traditional baseline correction and high-pass filtering suffers from many of the same potential pitfalls in terms of artifacts. Also similar trade-offs. Longer baseline intervals and stronger high-pass filters better correct for some types of noise, but at the increased risk of additional artifacts. Conditions are, if you use, following the publication guidelines, if you use baseline corrected data, you should also show the original ERPs together with the baseline. And even a more elegant way would be to include the baseline into your statistical model, suggested by Ale. I really highly will read this paper as it shows very aspect of baseline correction. So what I suggest is that to include the baseline into your statistical model, which is called the regression-based baseline correction. And how does that work? At the heart of EEG, but also fMRI analysis, there is the general linear model. On the left side, you have your outcome variable, y, which is usually the EEG activity. And on the right side of the equation, you have your predictor, which can be 2 different conditions or group or a continuous variable, and the error term. So now for baseline corrected data, we can rewrite this y into the y of post-stimulus activity, which is called here window, minus into the typical baseline correction. And now we have the new formula here. And we can now move this baseline term, y baseline term, on the other side of the equation. Because it's now on the side of the predictors, we can also rename the y baseline to x baseline. And now we have included the baseline interval into our statistical models. We can now even include an interaction term and see whether the baseline actually varies between different conditions. And this has the benefit that the regression-based baseline correction explicitly models the influence of the baseline instead of mixing the signals. And this does not strongly systematic difference assumptions that there is no difference between baseline intervals. And however, not eliminate the need for appropriate high-pass filtering. And you should be careful that you always apply this on single trial data only and on regression approaches. But this is considered as best strategies anyway. You should any average and statistical analysis. You should rather use the single trials into your statistical model by using mixed model and include the subject as a random effect. And this is also done Cyril Pernet's nice toolbox, the LIMO EEG pipeline. And Cyril also mentioned the regression-based correction in the future release. So now let's come to the summary of the filtering in ERP analysis. It's really difficult to give any general recommendation, as the filtering effect depend on the data, the signal-to-noise ratio, the noise characteristics, and the parameters of interest, whether you're interested in the amplitude, in the onset, or the latency. However, the goal of filtering is always to improve the signal-to-noise ratio. So if there is no noise, there is no need for filtering. Check out your raw data appropriately. And applied filters must not introduce distortion or artifacts systematically biasing your estimated. For low-pass filters, it's less problematic compared to high-pass filters because the higher frequencies have smaller amplitudes. Thus, cutoff frequencies higher than 40 Hertz are preferred. For high-pass filters, the distortion might be much of a bigger problem. We recommend here lower cutoff values in the range of 0.001 or 0.005. Detect filter distortion. Systematically explore the effects of different cutoff frequency, but do this only in your pilot data, because otherwise it's prone to p-hacking. So you take your pilot data and test different cutoff frequency and optimal cutoff for the real data, and make sure that the real data and the pilot data are recorded in the same noise environment, and inspect the different filtered and unfiltered data. Replace traditional baseline correction with regression-based baseline correction, as I just showed you. So let's talk about the effects of filters and automated ICA classifiers. Automated ICA classifiers such as MARA and IC-label gained a lot of popularity in recent years, and for good reasons. Because on the one hand, it takes out the subjectivity of your analysis and thus increases the reproducibility of EEG analysis. And you don't have to manipulate if this independent component is artifactual or not. And on the other hand, one of the big issues in neuroscience is that our studies are usually undersampled, and thus we need much larger samples. And it's even less feasible to do reliable and good manual artifact IC identification on large data sets. Thus, we need automated ICA classifiers. However, they carrots, which I want to show you now in the next couple of slides. So first of all, I first checked the effects of high-pass filters on the ICA decomposition. So I took the G and applied different high-pass filters and looked at the ICA decomposition. And here you see the results of it. I always plotted the first 10 IC dependent components. And what you have to consider is that the ICA is biased towards high amplitude. And remember that, that the EEG power spectrum shows us that the low frequencies have much high amplitude. So low frequencies have most influence on the ICA decomposition results. However, you're usually interested in frequency from something between 3 and 20 Hertz. And thus, it is recommended by Winkler, but also by Makeig here in the link, to compute ICA on EEG data, which are at least 1 Hertz high-pass filters. And we can see this also here in the analysis of the Wackerman and Hansen dataset. When we use cutoff frequencies lower than 1 Hertz, the ICA decomposition is not as ideal than when we use ICA with high-pass filters of 1 Hertz or 2 Hertz. The low-pass filter doesn't change much. But then we have the problem that when we use a 1-pass filter, this causes a distortion in our ERP analysis, showed you in the previous. So the solution of this is actually using temporal filtering, which I will show you right away. So, but now let's take a look first at the effects of different high-pass filters on those ICA automated classifiers. So I took the and applied different high-pass filters, computed the ICA decomposition, and then I ran IC-label and also MARA. And you can find the code and the results also here on the OSF link. And I used as an IC label setting, I used to exclude the components if they are labeled with a probability of 80% as muscle, eye, heart, line noise, channel noise, or others. And then I simply replotted the retained independent components. Again, always the first independent components. And what we can see here is that when we use a high pass which is lower than 1 Hertz, we have some components which still seem to be eye artifacts, or for example, hard artifacts. And you see also the label of IC label, it's still the most prominent label is eye or hard. And they just didn't exceed this 80% probability, and that's why they were still included. However, I'll argue that you could simply lower the threshold, but this comes at risk by also excluding components that actually contain some interest. Thus, it is recommended specifically for IC label to use 2 Hz high-pass filters and 40 Hz low-pass filter data for IC label. Why? Because the data set which was used to train the IC label classifier filtered with those filter settings. Mara uses a slightly different filter settings, but also from 1 Hz to 30 Hz. It's recommended to use the same filter settings as the training data. But then we still have this problem that if we use a 2 Hz high-pass filter, this causes our ERP analysis. So again, the solution here is temporary filtering. So what is this temporary filtering finally? The idea of this temporary filtering is that we create 2 versions of the raw EEG. And the one is actually temporary filtered for 2 Hz to 40 Hz, and then an ICA is applied, and then we conduct IC label and get the retained independent components. At the same time, another version of the raw EEG, we apply high-pass filtering with much lower cutoffs, which is seen here. Actually, it's way too high. So, and then we use from the retained independent components, we take the ICA weight and sphere matrix, apply it on this high-pass filters with lower cutoffs, and then we get the final independent components and the clean data. And here I wrote it out again for you to check it out later. And this applying the ICA weight and sphere matrix can be easily done with this EEG check set function in EEG lab. And here you see the results with the temporary filtering. You see that actually many of the independent components of the first hand, they are labeled as gray. And I would always recommend to do some using those automated ICA classifier as they are provided by IC label, which show you definitely as they provide you nicely the frequency spectrum, the time course, the topography, and the labeling. And I also did the ERP analysis with different high-pass filtering before the ICA and also with temporary filtering, which is also provided an OSF, which you can check out, but also the results. And you see if you apply temporary filtering to afterwards high-pass filters with a low cutoff, the signal is nicely retained. And you also see the effects for the ERP differences between famous faces and scrambled images. But if you use too high cutoffs for high-pass filters, you get distortions here, but also diminished effects. So now I would like to talk a little bit about the effects of multivariate analysis and high-pass filtering. Multivariate pattern classification analysis, PA, has become increasingly popular in EEG. But high-pass filtering potentially affects the decoding accuracy. This is particularly problematic in paradigms with long trial duration, for example, in working memory experiments where we have a long retention time. And the authors suggest an alternative method, which is called robust detrending, which in his paper, which is very accessible. And they simulated, or they actually conducted a test with different high-pass filters seen here on real data. And what they observed was that when you use a high-pass filter, which is 0.5, that you suddenly can decode whether a subject is remembering those correctly or not before the stimulus was presented. And this is definitely not possible. So those are some spurious artifacts filter. And they also smear the decoding accuracy over time. And if you use the robust, no such spurious effects of the decoding are shown. They also used simulated data to provide and to prove this effect. Because of the real data, you cannot really tell whether the improved decoding accuracy is real or not, but they also showed the same problems with real data. So if you use an MVPA, use high-pass filters with other frequencies for MVPA analysis. So finally, I want to introduce our standardized pre-processing toolbox, Makeig. The motivation to invented Makeig was actually that we are looking for biological measures for psychiatry and neurology. To do this, we need very large data sets. And also those large data sets, they are collected over years. So we need a toolbox where we can make sure that the same pre-processing is applied even for data sets which are coming in later. But you can also use Automagic for small data sets. And Automagic provides us standardized pre-processing, which gives you reproducible results and takes out any subjectivity out of it. And it's very well done. Here you see the workflow of Automagic. So it starts with the raw EEG here, seen in yellow highlighted. And first, we apply some bad channel detection with state-of-the-art, such as prep or clean raw data. And what we want to highlight, also use temporary filter to identify the bad channels, which is easier. And once we identify channels, we remove the bad channels, and then we offer different filters. We offer the default EEG lab filters, but you can also create your own filters. And we offer also a clean line to remove the power line noise. Then we have implemented different automated ICA classification algorithms, such as MARA and IC label. And we also provide here this temporal filtering for the automated classification. And there's also optional robust PCA, which we usually don't. And all of this is very modular. So you can choose the combination of the different pre-processing steps by yourself. And what is very important is that we also offer quality measures, objective quality measures. So far, we have 4 quality measures. We also plan to include more. But the benefit, you don't have to subjectively decide whether a data set is good or not and should be included or excluded. But here you have actually objective values based on which ones you can set some thresholds and report them also in your paper. This makes it very reproducible. And we also provide some data visualization to see the different effects of the individual pre-processing steps. And then we provide also a log file of the methods, which helps you to write the method section correctly for your publication. And it's bits compatible, so it can load data which are already in the bits format, but it can also export the data into the format if you wish. And currently, Automagic is available as a MATLAB toolbox, but also available as an EEG lab plugin. And we are currently working on a Python version, which we call Pythomagic. And the code is available here on our GitHub page. We also wrote an extensive wiki which explains all the individual steps and help you to guide through Automagic. The take-home message is from my talk is that filtering results in distortions of the temporal dynamics of the signal. For ERP, means that high-pass filters systematically bias signal onset latencies and signal amplitudes. For MBPA analysis, high-pass filters potentially affect the decoding accuracies. Thus, filter as little as possible. However, this is often not feasible as the data is contaminated by artifacts, and ICA, for example, requires high-pass filtering. If filtering is required, use sharp high-pass filtering 001 and 01 hertz, and substitute pass filters by successive shallow low-pass filters and sharp high-pass filters, and substitute notch filters with clean line and zap line. And filters should be always on continuous EEG data, not on epoch EEG and on average data. As I showed you, those effects, they are, again, always calculated on continuous EEG data. Distortions on epoch EEG data is even much bigger. And systematically compare the data on frequency cutoffs, but always only on pilot data, otherwise p-hacking. And rather use regression-based baseline correction in traditional baseline. And also consider temporal filtering for bad channel detection in ICA automated classifier, as we also implemented it in Automagic. And record correct and accurate filter parameters and report the quality. And as we provide them in Automagic, and ideally you use Automagic, so you have the correct workflow for pre. I would like to end my talk with the quote from Wittmann, which says, "Filters are like a sharp knife, a very useful tool, but to be handled with care." All right, that was it. I hope you enjoyed it, and thank you for listening.
All right, so hello everyone. So my name is Alex Gramfort. I'm a researcher at INRIA in France, in Paris. And today I will do this presentation for the educational course on EEG, MEG, and aspects to pre-processing. And so the title of my talk is Data Normalization, its impact on source modeling and multivariate analysis. So I insist on the word multivariate, which is really a core aspect of what I would like to teach you today. So before this, let me start by making this statement. So when you process EEG or MEG data, you have a fairly, I would say, long list of things to do to your data. So it's what I would call a chain of steps, a pipeline. And here, what I would like to illustrate here is that you should be careful to every step of your pipeline, because if one of your steps is not correct, then you will screw up your entire pipeline. So pay attention to every aspect and check that your steps are valid and proper. So a few things before going into the details. You will see in my slides, sometimes you will see the acronym THM. So what it stands is take home message and not theorem. And here, if you see this Jupyter logo, it means that there's a link and a page that you can visit, and where you can download a Jupyter Python notebook in order to replicate the figures or dig deeper. So here, what you see now is a page of 10 seconds of actually MEG data. Here is many lines. Okay, you have many lines because EEG and MEG data are multivariate. What multivariate means is that you're not observing a single time course, a single signal at the same times, but multiple ones. So here, I would say in EEG, you go from, I would say, a few dozens of EEG sensors to 100 or even more if you have a high density EEG system these days. Or MEG, I would say standard these days would be to have about 300 MEG sensors. So you really have a lot of simultaneous signals. It's really important to take care of this and to treat these signals globally. And see here, I'm also illustrating with these slides that in such data, you have artifacts. So we talk about pre-processing. Here you see drifts. You have these signals that are moving slowly with slow frequency, slow waves. You see all these high frequency noise that you see on the signals, which are the landline noise. You have these peaks at roughly every second that correspond to the electrophysiology artifacts due to the heart of the subjects. And sometimes you also see blinks, which is also something which is due to the subjects in the machine. So let me start with something quite basic in order for you to illustrate and understand fully what's going on here. I insist on the fact that here we look at multivariate models. So here, consider the problem of having two EEG signals. I'm going to call these signals X1 and X2, and I'm going to assume that X1 is 2 times a signal that I'm going to call S of t, plus some additive noise, which I call N of t. And now suppose that X2, which is another EEG signal, is just equal to the signal ST plus the noise NT. And S and the source and the noise are assumed to be the same and common between these two EEG sources. Okay. Now I'm going to do something rather simple, which is if I just subtract my two signals, actually I should subtract X1 from X2 to make this work, but sorry about the tiny mistake. But here what you see is that by subtracting the two signals, you do get rid of the noise. Okay, so it's a very simple step. It's just a linear combination, which is here actually just a subtraction, where I get a perfect estimate of the signal by just treating the signals as multivariate, because I will process two signals at the same time, and I will basically leverage the fact that I have two signals in order to get a better estimate of what I call here the SNR, which stands for the signal to noise ratio. So this is a very simple example, and now I'm going to illustrate this with more involved examples. There's a notion called signal subspace projection, which is a linear combination of sensors that you subtract to the data in order to denoise them. So what you see here on the left is real data, okay, without any projection applied. As you see that you have some, I would say, rather noisy signals, and also you can imagine that you have slow drifts in them. And now if you apply some kind of linear projection, so linear combination of sensors, I will reduce here these artifacts which here are due to environment. And you see on the upper right corner of my slide, this idea of when you do this, it's projecting your observation on this plane, which is here, this green plane that contains a better signal. And you can also do this with physiological signals. What you see here is that on the left, lower left, you have the heartbeat that appears. And if I apply a projector, I can get rid of my heartbeat by just doing this linear combination of sensors. So again, what this shows you is by using the fact that you have multiple signals, you have a multivariate data set, you can remove both environmental noise, but also physiological noise. At the bottom, you have a slide, a link to a page that you can visit in order to replicate these figures and learn more. Now, I'm taking a slide and a figure actually from a paper from Stefan Haufer and colleagues, where this shows you that in the context of brain-computer interfaces, these ideas of using multivariate nature of the signals is extremely frequent, extremely common, should be more considered in the context of cognitive neuroscience and when doing EEG. So here, in order to illustrate this, imagine that you have a subject that is basically thinking about either moving his right arm or moving his left arm. Moving the right arm would lead to these blue measurements that you see on channel X1 and channel X2. And when he's thinking about moving his left arm, what you obtain is these red measurements that you see also on the axis. What you see here, if you project these blue dots or red dots along the X1 axis, you see these distributions, okay? And this distribution that you see in blue and red, you see that they are partially overlapping, but not too much on the X1 axis, okay? Now, if you see on the other axis, which is X2, now the distributions are almost perfectly overlapping, which means that if you were to try to disambiguate what is the right arm or the left arm, by just having a single sensor and doing a statistics only on channel X2, it will not work at all. It will work slightly better if you look at channel X1, but actually what you should do, and what is done by techniques, for example, like linear discriminant analysis, you would find a way to linearly combine your sensors in order to find this oblique line that maximally separates your blue dots from the red dots, okay? So this is really what is illustrated here. In order to increase your statistical effects, in order to increase your ability to statistically differentiate your experimental conditions using EEG on MEG, it's a very good idea to combine sensors and to extract signal from. Again, at the bottom, you have this link. You can click on this link and you will go to this page where you can play with these ideas. So again, take a message, by doing linear combination of your sensors, you can improve the signal to noise ratio. Now, let me talk about another aspect where the fact that you have multivariate data is extremely important. So this is related to the question of source modeling. So what you're trying to do when you do source modeling is to try to find what is happening inside the brain from data that you record here on the scalp. So what you see here is an EEG topography. And what you're trying to do here is to find what is happening inside the brain. So here you have this field of potential on the skin. And again, you're trying to find what's happening inside the brain. So in order to do this, let me try to illustrate a few concepts. So what you see on this is what's happening from an EEG or MEG perspective when you put a source inside the brain. So here it's a source put roughly in the left auditory cortex at the fixed location by changing its orientation. So what you see on the top row is MEG data, on the bottom row is EEG data, and you're basically trying to move, I mean, not move, but really rotate a source inside the brain in order to produce signals, okay? And you see when you see it's red, it means that you have a positive field, so positive magnetic field, or at the bottom, a positive potential with respect to a reference. Okay, so what I would like to understand, to explain to you on this slide is not really too much the physics of MEG and EEG source imaging, although it's quite interesting. It's not really the topic of this course today. What I would like just to explain to you is that basically the physics, which is due to Maxwell equations, allows you to predict what you will measure on MEG and EEG given a specific source inside the brain. Okay, so this is what this take-home message tells you. We do know a model that can predict what are MEG and EEG data given a dipole inside the brain. So now comes the question of source imaging and dipole fitting. So here, imagine you have your multiple MEG sensors or EEG sensors, you have your multivariate data, and what you see here on the right is just a cartoon picture using the Earth and various satellites. And in the end, you can imagine that when you do dipole fitting, so you're trying to fit a dipole, meaning that you try to fit the location and orientation of a dipole that will explain your data. You're kind of doing some kind of triangulation. You have your attenuation of the field given the distance between the source and the measurements. And given these attenuations that you measure at different locations, you can pinpoint the location of your source. So now, how would you solve this practical problem? Well, you can use this rather brute force idea, but at least it gives you an idea of how to proceed, okay? So the idea that you can propose is basically try to test every possible location inside the brain for every possible orientation and take the best. Now the question is the best in what sense? So here what you see in this formula that is circled or highlighted at the bottom left, you say that the estimated R, so the location and orientation of the dipole, is the location for which you have the minimum error in the Euclidean norm sense. This error is just the norm, the squared norm here between the actual measurements in EEG and the forward model. Okay, so the propagation of the field given the dipole, a source at location R. Okay, so here your measure of goodness of fit, your measurement criteria, your error measurements is just the sum of the squared errors. Okay, so now you have your criteria to select what is best. You can test potentially every possible location and orientation, and you say, okay, now the job is done. Let's see now in practice what's happening when you do things like this. Well, what you see in this slide is median nerve stimulation data, and you're trying to locate inside the sensory motor cortex what is the location of the dipole that is active following this medial nerve stimulation. The first row corresponds that when you use only 7 sensors, okay? The second row is when you use 42 sensors, and the third row is when you're using 92 sensors. And then every column is for a given time instant. So if you compare the different rows, what you can see is that if you go from top to bottom, the size of the circles that you see here in yellow, these circles are these confidence regions. So you can see these as confidence intervals, but in 3 dimensions, these confidence regions that tell you where the dipole fit solution is believed to be. So what you see is that when you use more sensors, when you use the fact that you have multivariate data, then you do get smaller circles, which means that you get more confidence in where the source is actually located. And then what you can also see when you compare now the different columns, what you can see is when you go from left to right, what's happening is that you look at different strength of the field on the sensor, so different strengths of the dipole fields, and it just tells you that if you estimate the source location based on the peak amplitude with the strongest signal to noise ratio, then you get, again, more confidence in where the source is located. So using more sensors is a good idea to increase your ability to locate with high confidence, and also SNR is a second variable that matters. So now I would say that the devil is in the details, and in order for you to understand this, I'm going to suggest the following thought experiments: that you have a sensor that is extremely noisy, and in the end, you would like to be able to do this dipole fit, even if you have this sensor that is there, that maybe contains signals, is just way more noisy than the others. What I guess you can understand is that if you try to find the best candidate dipoles, and you try to approximate every sensor equally well, if one of these sensors is very noisy, then you're kind of shooting yourself in the foot. You should not give the same amount of credit or care as much for the sensors that are more noisy. So now I'm going to take a few minutes to introduce you to the concept of inverse modeling, to tell you where this notion of noise between sensors is present, and also give you some suggestions about how to address in a mathematically principled way these issues. So what you see here is on this slide is just to introduce what I will call the measurements. This is just a matrix that I call M, where every column of M is a topography, so a field on the sensors, and every row is a time course. What you see here in this slide is now X. So X is the notations for the source amplitudes, okay, where things are happening inside the brain. This is what you're trying to recover. This is the where and the unknown source location that you're trying to recover. Okay, so X is what I call for my unknowns, which are going to be my source estimates. So in order to do this using a distributed source model, okay, this is an alternative to the dipole fit. What you would do is you will discretize your source space using here all of these yellow dots. And for each of these yellow dots, you're going to compute the forward model that I illustrated before using these topographies on the left using EEG or on the right using MEG. So you're going to compute the forward model for each of these yellow dots. This will give you different columns of the matrix G. And from this entire matrix D, then you can solve the inverse problem using a distributed source model. Okay, so the lead field here, the gain matrix or the forward operator for the EEG and MEG is just the concatenation across rows of the two matrices. So once you're there, you can exploit the fact that you have the physics of Maxwell and which relate the unknown sources X with the measurement M through this matrix G that we just mentioned and that was computed using the forward model. Okay, so in mathematical notation, you have your data M, which is equal to the dot product between your gain matrix G or lead field matrix G times the unknown sources plus E, which stands for the noise. So once you've put these notations, when you try to exploit all your multivariate data M in order to find the sources inside the brain, you will write this here what people call the minimum norm solution, or something that looks with the minimum norm solution, depending on the choice of the phi function, the regularization. Well, the topic of today is not to care too much about regularization, but to insist on here what I call data fit. So data fit is the first quantity that you see on this equation that is here circled in red. And here M minus Gx is this quantity that should be small if your job is done properly. So X is your unknown that you're trying to estimate, and you're trying to find X such that M minus Gx is small according to this norm, which is here the Frobenius norm that is here mentioned. So the Frobenius norm of a matrix A, okay, so if you square it, it's just the sum of all the entries that you see inside this matrix. So the Frobenius norm of A squared is just sum over ij of the Aij squared. Now, imagine or remember that this matrix M can contain MEG, EEG, and also sensors with various noise levels. So the question is, okay, now I want to leverage all my multivariate data. I want to work with all my sensors at the same time. How do I put them in the same scale? How do I normalize them? Or how do I whiten the data, which is the technical term to do this, in order to have this principled formulation? In order to illustrate this, let me show you some actual data. So what you see here is at the top, gradiometer data, so MEG. In the middle, it's the EEG measured simultaneously. And at the bottom, it's magnetometer data, which is also here measuring the magnetic field. And you see here that on the top, you are measuring femtotesla per centimeters. In the middle, you're measuring microvolts, okay, in EEG. And at the bottom, you're measuring femtotesla. So in some sense, you have like apple, bananas, and strawberries, three quantities of 3 units, and you're trying to combine them and normalize them or scale them properly. Okay, so how are you going to do this? First, you need to look at what we will call the correlation structure or the covariance structure between all of your data. So here, these covariance matrices are quantifying how much 2 sensors are varying together. If a quantity is positive, okay, it will mean that these 2 sensors are positively correlated. If the quantity is negative, it's negatively correlated. So here, this gradiometer matrix that you see on the left, if you look at the diagonal of this matrix at position I would be the variance of this channel index I, okay? What you see also in this matrix is that this matrix is clearly not a diagonal matrix for the gradiometers and even less for the magnetometers. So you do have a correlation structure between sensors, but you also see, for example, in the magnetometers in front of you, so around channel 50, you have this big red square, which suggests that you have one magnetometers or a few magnetometers that are a lot more noisy than all the other channels. Okay, so the fact that you have different signals strength or the signal noise strengths across sensors is actually present in empirical data. Now, how do you estimate this covariance from data in order to compensate for the fact that you have different noise levels? Well, the problem is that if you just try to apply this formula that you see at the bottom, C equals just the M times N transpose divided by the number of time points, it's not always a good idea. So here, covariance are also present in beamforming, which is another very popular way of doing source imaging using EEG or MEG. What you see in this figure taken from a paper from Mark Woolrich and colleagues, which is that if you only use 10 seconds of data, which is the first row, and 80 seconds of data, which is the second row, you get different pictures. And what you see on the different columns is the depending on what parameter mu you're using, and this mu parameter appears on the formula that you see on the bottom left. So when mu is equal to 0, you recover the true empirical covariance that I showed before. And if mu is strictly positive, then it means that you're biasing your covariance by adding a tiny identity matrix on the diagonal. What you see by looking on the upper left is that if you don't set any mu, so if mu is equal to 0 and you don't have a lot of data, then your results look not very nice. Now, if you regularize with a mu equal to 20 on the right side, if you use 10 seconds or 80 seconds, you have a roughly poor spatial resolution, and you see a big blob inside the brain. But now if you have 80 seconds of data and you don't regularize too much, then you have a very clear picture, okay? So what it means is that this covariance, depending if you use a true empirical estimate or if you regularize it, you get a different picture. Well, it turns out you have different ways of estimating this covariance matrix, which really try to capture this at least one ways of estimating empirically these covariance objects. I'm not going to go into details. What I showed you before is with Mark Woolrich work. is the option 1 when you add a tiny constant to the diagonal. But you have another approach which is called the Dwarf, which is used across many different applications, including in brain imaging, in BCI, EEG, also fMRI. You can also try to use a probabilistic approach like PCA, probabilistic PCA, that assumes that every sensor is corrupted by a different, same noise level, sorry. So PCA would be the same noise level across sensors plus a low rank structure, HH transpose, or factor analysis, which will assume that you have also a low rank structures, H and H transpose, plus a different noise levels between all sensors. And then what this equation in this slide shows you is that actually you can test using empirical data if your estimation of the covariance is correct. You can just test this by looking at the likelihood of your data according to the hypothesis that your data follows a Gaussian distribution with parameter, with covariance C. Okay, so if I call this covariance C my believed covariance about my data, I can look at the likelihood of Y, the likelihood of my data Y, given my matrix C, for which you have this closed form. In some sense, it's not very important, this formula and the detail, it just tells you that if you have a statistical hypothesis about your data, you can usually test if this hypothesis is correct before going further and pursuing in your analysis. So here now I'm going to illustrate this with real data in order to show you that these mathematical concepts can be visually checked using simple plots. So for this, we did some experiments using various data sets. We used many MEG data, but here I'm going to also show you, I mentioned we tested using one EEG data set. And here, let me try to illustrate. Once we take into account the covariance structure of the data and use this covariance structure to whiten or to scale the data according to this estimated covariance, what we obtain is basically whitened data or normalized data. Remember the title of this presentation, which is how to normalize data in order to to make the best of EEG or MEG data. So here I've normalized my data in order to make sure that my data are all Gaussian distributed with a zero mean and a unit covariance, an identity covariance. And this is what you see here. If you look at what's happening before time equals 0, you have these two horizontal lines, which are at minus 1.86 and 1.96, which are basically the confidence regions, or basically the interval, in which if you have a Gaussian distributed variable, 95% of the value should lie. So what you see here is that by doing this pre-processing, this scaling, you've managed to make sure that your EEG data, all the channels have roughly the same noise, and they all lie inside this interval. Okay, so it's also a good way to check that during baseline, your data is really matching the statistics of the noise. And then if you deviate from this, I would say, band between these two red horizontal lines, then you start to see signal appearing according to your noise estimate. And then you can also look at what people call the global field power after you've whitened the data. And mathematically, what the statistics tells you that once you do this, you recover a chi-square distributed variable, and in order to, and this is what you can actually test. So if you've done your whitening properly, if you've done your pre-processing properly, exploiting the multivariate nature of the data, then the lines that you see here, color-coded, should be roughly around 1. Okay, so there's a reference at the end of this presentation where you can look at the details in order to learn more mathematical details about this. Okay, but the take-home message is that you can make these simple plots in order to check that your hypotheses are correct. In the end, once you've done this, you can produce these types of images, which are source estimates here using MEG and EEG data, okay, combined, using a minimum norm. All right, now I'm going to show you that these hypotheses, when you check them and they're valid in sensor space, they produce more meaningful results when you look into a source space. So what you see here is that if you only use 20 epochs, which is the first column, or if you use more epochs when you go from left to right, basically by using a proper estimate of the noise and a proper scaling and proper whitening, then on the bottom row, you see that the brain maps look reasonably similar across conditions, given if you have 20 epochs or more, whereas if you only use 20 epochs and you don't pay attention to the statistical control of your noise normalization, then you see this massive activity inside the brain, which is very spurious. Okay, so now I'm going to switch to an example using a Jupyter notebook before concluding this lecture. So for this, I'm going to use this data set that you can get access to on the Brainstorm website. This is actually phantom data. So this is going to be MEG data recording using a realistic plastic phantom that allow you to obtain real data using a ground truth location of the source. Okay, so it will allow us to quantify on when we know the ground truth, how much checking our hypothesis about the noise matter in terms of source localization. So this is using MNE, the MNE Python package in a notebook. I invite you to visit the MNE website to learn more about this. So, but here I'm just going to walk you through this notebook so you can understand what's going on. Okay, so I'm going to import the necessary dependencies, the necessary libraries, using here NumPy, Matplotlib for making the plots, and then MNE imports. MNE comes with many data sets that you can easily fetch from the web in order to replicate our results. So here I'm going to fetch the data. Okay, so my data is a NeuroMac MEG file, so it ends with the fifth file. So I'm going to use read row fifth. And read row fifth will give me a row object that I read, contains the continuous recording. Then inside these continuous recordings, I have a stimulation channel that tells me when the dipole of the phantom is activated. And this dipole is going to be activated, we will see, with the sinusoidal signal inside the phantom. So this is your continuous recordings, all the MEG signals. And you can look at the power spectrum density, so the PSD of your data easily using row.plotpsd. And here you will see with every line corresponds to the power spectrum density of a sensor that you see here represented on the helmet. Okay, our phantom produces this sinusoidal burst that we'll see in a minute. So here this is plotting the data using the events. And now I'm going to define epochs, which means that I'm going to extract chunk of data around the onset of the phantom activation. So for this, I obtain my events, which tell me when do I have these onsets. I have many dipoles that are actually present in the data. I'm going to consider 32 of them, from 1 to 32. And here, what you see here is just the evoke response, so the average activity of the phantom for the location of the dipole at position 1. And here you see that during baseline, so before 0, your signal are rather flat, which is good. And then at time equals 0, then you have your phantom that start to oscillate. And you see that it takes a bit of time to ramp up, and then it stabilizes to make these regular sinusoidal patterns up to 100 milliseconds. So since the phantom is a spherical device, I'm going to use a sphere model. So this is what allows me to compute a forward model in the Maxwell sense. And here, what you will see using this plot alignment, this will produce this image that you see actually here. This image shows you here the sphere, where the phantom is located, and here the MEG helmet. So each of these square here on the helmet is an MEG sensor. Okay, so now I've positioned my sphere, I've got my sensor, and here I'm going to compute the covariance from my epochs using the baseline data. So here the baseline is going to be here, this period that you see here, And here, what I'm going to use is this M&E function called plot evoked white, which will show me these sanity check plots to make sure that if I whiten my data using this covariance that I just estimated, things go fine. And what you see here is that it's really, really good. What you see is that after you whiten the data, then your whitened global field power is around 1, which is exactly what you anticipated and what you hoped for. Okay, and then you see that the signal goes stronger when you have the dipole that are active. Okay, so this is really something that you should look for in your data to make sure that your scaling and normalization of the data has been done properly. Now I'm going to fit some dipoles inside the brain at a given time. So here I'm going to use 36 milliseconds, so roughly around here when you have this peak. And for each of the 32 dipoles, I'm going to fit a dipole on the data after whitening. Okay, so after scaling the signal properly. And what you see here is that, we've actually estimated properly. So here it's a residual plot. So you see that there's not much left after you've estimated the location of the source. And here, I'm going to plot here the errors in terms of localization, okay, in terms of orientation, and in terms of amplitude strength. What's important is what I illustrated here is that the errors are less than 5 millimeters, okay? And for some sensors, it's very, very much less than 2 millimeters. So it's really, really good, okay? So now, at the end, you can visualize here where have you estimated the dipole. So what you see here, you have two colors. One color is the true location of the dipole, and the other color is where the dipoles are actually estimated. And you see that it's pretty close. It's pretty good. Okay. Now I'm going to do something a bit different. I'm going to use a covariance structure, which basically discards and ignores the fact that you have different sensors and sensors which are corrupted by a different noise level and also correlated. So here I'm going to use this ad hoc covariance, which is just a diagonal covariance that ignores, again, the correlation structure and the different SNR across sensors. So let me just do this. What you see is by running this code that suddenly your line starts to go beyond your -2, so minus 1.96 or 1.96. Okay, you see that you're not anymore around 1. And now let's see what's the impact in terms of localization of my dipole. So here I'm going to refit all of my dipoles. Here you can look at the residual, but what's going to be particularly striking is actually the location of all your sensors. And what you see here is that you have some dipoles for which you make an error which is more than 3 centimeters, okay? That you see many dipoles for which you make an error on the order of 1 centimeter or more, okay? And just by ignoring properly and ignoring the fact that you have a noise structure. And here I'm going to now show you inside the helmet. Here, now it's the same image as before, and you can see that you do make some big errors in some locations, some of these errors that you could not see before. All right, now let me go back to my slides, and I'm going to conclude. So what I just showed you is basically this example where we've looked at this phantom data where we had these small errors with the proper covariance. And now if we use an improper covariance structure, an improper normalization of the multivariate data, then things are much worse. And now I'm going to conclude. So what I try to convince you with is that if you don't pay attention to the data and the statistical hypothesis, what I call this garbage in, garbage out, then you should not trust what's happening at the end of your pipeline. Something I would like to stress again and again is that MEG and EEG are multivariate data. So stop looking at EEG and MEG and MEG sensors by looking at one sensor at a time. You're not helping by doing this. And again, check your statistical hypothesis and use quality control plots and metrics in order to make sure that you're on the right path. With this, I will conclude by just give you some references to the work of my team. So this is a paper that we published a couple of years ago showing you how to apply all these good practices using a publicly available data set. So if you go to this page, you will have a description, and also it links to code that you can use on your own studies. This is the paper published with Dennis Engerman, where we explain the details about how to normalize this data using this covariance. So if I was a bit too fast on this aspect, please have a look at this paper. And if you want to apply this to your data, you can visit this M&E website from where I took all the examples that you've seen in this presentation, and that you're very welcome to use. And if you have any question, you can reach us on the mailing list or on GitHub. All right, thank you very much. And thank you very much for all the funding agencies that have helped me with this work over the last years. And that's it for me. Thank you. Bye-bye.